Assalamu alaikum everyone. I'm Zainab Nawaz, um, an undergraduate from University of Sri Jayewardenepura. So today we'll be going through the lesson genetics. So when it comes to genetics, the first scientist to give a a huge idea about the inheritance was Gregor Mendel. He is considered the father of modern genetics. Mendel discovered the basic principles of heredity by breeding garden peas in a carefully planned experiments. So while he was conducting the experiments, the chromosomes were still not identified. So his experiments and discoveries were entirely done, deducted using the experimental findings of the garden peas. Later on, after the discovery of the electron microscopes and other microscopes and development of the cytology, these chromosomes were identified. So before we start with the genetics, we have to know certain terms in genetics. So among that, the first one would be character. So character is a heritable feature that varies among individuals of a population. So this is something like blue eyes or black eyes of a person. Here, the color of the eye is the character that varies among individuals. So there could be lots of individuals with different colors of eyes, and the character will be the color of the eye. And there is a similar feature called trait that is the variable of the character. So if we consider eye color, the colors are traits. Black color eye is a trait. Blue color eye is another trait. So the heritable variance of a character in an organism is a trait, while the heritable feature that varies among the individuals is character. So if we consider the garden pea, there could be different characters such as seed form, cotyledon color, pod shape, cotyledon color, flower position, and so on. And they would have different traits. So if we consider flower color, there could be two different colors, purple and white. And then we have something called gene. So gene is the basic unit by which genetic information is passed from parent to offspring. So this is a nucleotide sequence of DNA present in the specific part of chromosome. So if we consider one chromosome, it would contain thousands of genes coding for different characteristics of that organism. And then we have alleles. So alleles are the alternative versions of genes. If we consider hair color, an individual would get two alleles, two genes from one from the mother and one from the father, and both of these are considered as alleles. So alternative versions of genes are called alleles. And if two different genes, two identical alleles are called as homozygous, while two different alleles are called heterozygous. So hair color from the mother and hair color from the father would be considered as homozygous alleles, while hair color from the mother and eye color from the father would be considered as heterozygous alleles. And then we have the genotype and the phenotype. Genotype is the genetic makeup or the set of alleles of an organism. So this consists of the entire genes that an individual inherits from the maternal and paternal side. So whatever the genes they receive from the mother and the father is considered to make up the genotype of the organism. As for the phenotype, that is the part that is only expressed. So if we consider an organism, the feature that is observable is considered as the phenotype. When we consider the pea plant, the flower color could, if we represent it as capital B for purple and simple B for white, the genotypes could be capital B, capital B, capital B, simple B, or simple B, simple B, whereas phenotype is always either purple or white. And then we have dominant allele and dominant trait. So dominant allele is basically the allele which is expressed. So when we consider this example, we have capital B and simple B at the heterozygous state. So in that state, which color would be expressed is decided based on the dominant allele. So at heterozygous state, the allele which determines the organism's phenotype by masking the expression of the other is known as the dominant allele, while the trait produced by the dominant allele is called the dominant trait. So in this example, we have purple flower color as the dominant trait. Well, also we have recessive allele and recessive trait. So recessive allele is the allele which is masked at heterozygous state, and recessive trait is the trait that is masked by the that is expressed by the recessive allele. So in this situation where we have capital B and simple B, the recessive allele would be simple B, which codes for white color, and the recessive trait would be white flower color. And then we have purebreds. So purebreds are offsprings obtained by self-pollinating over many generations. So offsprings obtained by self-pollinating over many generations, they would have the same genotype over the generations. So these are pretty useful in experiments, especially in Mendel's experiments. He used most of these purebred plants for his experiments. These uniform lines produced from self-fertilization of pure breeding varieties over many generations are called pure lines. And then we have hybridization, which means mating or crossing of two pure breeding varieties with contrasting traits. So if we go to the earlier example, if we consider a purebred purple flower color plant and a purebred white flower color plant, if we cross those two breeding varieties, we would get a hybridized plant. And then when we do such a cross matching, the parental plant is known as P generation, while the first generation of the offspring is given by F1 generation, or the first filial generation. And then if the progeny that results from the self or cross-pollination between these F1 generation plants, they are known as F2 generation. And then an organism that is heterozygous with respect to a single gene of interest resulting from a cross between parents having homozygous condition for different alleles of specific gene is referred to as monohybrid. So if we consider the flower color, where the purple color is represented by capital B, and if the white color is represented by simple B, the offspring that is heterozygous with respect to one gene of interest, resulting from a cross between parents having homozygous conditions for different alleles of a specific genes, is known as a monohybrid. Whereas a dihybrid is an organism that is heterozygous with respect to two such genes. So earlier we have one gene, and if we are considering two purebred plants that have purebred lines for two characteristics of interest, maybe flower color and the rounded and wrinkled nature of the pods. We could have rounded and wrinkled nature, and the organism, the offspring that results from the cross would be a dihybrid organism. And then we have test cross. Test cross is the breeding of an organism having unknown genotype for a specific dominant trait with an organism having homozygous recessive condition for the same specific trait to identify the unknown genotype. So this is when we have a plant, if we consider a plant having purple color flowers, we do not know whether it has homozygous genotype or heterozygous genotype. So to identify this, we cross this with another homozygous recessive condition organism, so that it would, the offspring from the offsprings resulting, we can identify the parental organism's genotype. Okay, and then we can start with the monohybrid cross. So Mendel derived his first law of inheritance by following only a single character in one breeding experiment. So when he started with a single character in mind, and did many experiments based on that. And then he counted the offsprings and the expression, their phenotype, and from the phenotype he could guess the genotype. So if we consider parental generations, he took two purebred plants, one of purple color flowers, and the other of white color flowers, and then he crossed them, and his observation was the first filial generation, the F1 generations, all had purple flowers as their color. And then self-pollination or the cross-pollination of the same F1 generation resulted in the F2 generation having purple color flowers and white color flowers in a 3:1 ratio. So this is not exactly 300:100. This is somewhat a little more or less, but since these are we consider this is a huge number, we ignore those small differences. So this was the actual number he got. From this, he derived the simplest ratio, 3:1. So through this, by repeating the same experiment for different characters, such as the nature of the pod and shape of the pod, color of the pod, shape of the seed, color of the seed, and position of the flower, length of the stem, and so on and so forth, he was able to come up with the theory of law of segregation. He came up with the first law, the law of segregation. So this law of segregation was put forward to explain the 3:1 ratio Mendel observed in his cross matchings of the F2 generation. So as per his hypothesis, each heritable character, so he considered the heritable factor. We have to keep in mind that during his time, the chromosomes were not a common concept. So he described his experimental findings as heritable factors. So he said that when purple color flowers and the white color flowers were crossed, the purple color flowers gave purple color heritable factor. They had the heritable factor purple color, and the white color flowers gave the heritable factor white color. And so when these two heritable factors during the formation of gametes, the alleles for a heritable character are separated and get into each of these gametes form. So that is the first law of Mendel's, which we call as the law of segregation. So he said that when we consider a heritable factor, such as if we consider purple color flower color, this character is determined by two heritable factors, which is really allele. He's talking about alleles. So if we consider purple flower color, he said that there are two heritable factors or two alleles that determine the heritable color, heritable factor. And then two heritable factors, and each of these are separated when they make up the gametes. During the formation of the gametes, the alleles for a heritable character are separated and get into each gamete's form. So that is the first law of Mendel, called the law of segregation. So we can also represent this using represent this using a Punnett square, where we consider the plant heights. So if we consider the parental generation, the parental plants to have parental plants of tall and dwarf plants, when we cross them, purebred plants of tall and short plants. The tall plants, we consider them to have the alleles capital T and capital T, while small plants, we consider them to have alleles simple T, and when they form the alleles, they form capital T, two alleles. And dwarf plants, they have simple T and simple T alleles. And when they form the offspring, they could form 4 different combinations. They could form 2 different combinations of offspring genotype. So this one allele from each parental plant would involve in forming the offspring. So both of these genotypes would be capital T and simple T. So though they are capital T and simple T, they are expressed as tall plants considering their phenotype. So this would be the F1 generation. And when we cross-match the F1 generation, when we consider the F1 generation, cross-matching of the F1 generation, we have capital T and simple T crossed with a capital T and a simple T. So these would be separated into gametes as capital T, simple T, and then capital T and simple T. So these alleles could form combinations like this, where the capital T from one parental plant and the capital T from another parental plant could form capital T, and one capital T and simple T could form capital T and simple T, while also simple T and capital T from the other one could form capital T, simple T. and then simple T and simple T could form the simple T. So these are three different types of genotypes that will be expressed as two different types of phenotypes. So the genotypes here would be capital T, and capital T, simple T, and simple T. But the genotypes would be, the phenotypes would be tall and short. So based on this, we can come up with the ratio 3:1. Here, this would be considered as a homozygous dominant plant, and this would be a homozygous recessive character, and this one is a heterozygous dominant phenotype. And when we consider dihybrid cross, Mendel considered two characters at the same time. So a cross between two heterozygous organisms with contrasting traits for two specific characters being followed is known as a dihybrid. So we can consider two factors such as yellow round seed, yellow color of the seeds, or green color of the seeds. Well, also we can observe whether they are round seeds or wrinkle seeds. So here the features considered are yellow and green, the color of the seeds. And we can also consider the nature of the seeds, whether they are round or wrinkled. Um, Sorry about that. So when we consider the dihybrid cross, we consider two characteristics of the same plant. So when we have to consider a purebred plant for two features. So the features we are considering are the seed color and the nature of the seed. So the cross, when he performed this experiment, he found out that the F1 hybrid plants had all yellow round seeds. So based on this experimental finding, he came up with the conclusion that yellow seed is dominant to green color of the seed, while also round nature of the seed is dominant to recessive wrinkle nature of the seed. So the F1 hybrids are heterozygous for the two characters being followed in the cross. If we give the allele combination capital Y for yellow and simple Y for green, and capital R for round, and simple R for wrinkle, then the heterozygous F1 generation showed the genotype capital Y simple Y and capital R simple R. So this is the genotype, and this would be the phenotype observed in the F1 dihybrids. So based on this conclusion, Mendel came up with two alternative hypotheses where they could be dependent assortment of alleles or independent assortment of alleles. So dependent assortment of alleles is where the two characters, even the yellow and the color of the seed and the nature of the seed, they segregate based on one another, or independent assortment where the segregation of this nature of the seed is independent of the segregation of the allele responsible for the color of the seed. So his first hypothesis could be dependent assortment, where the color segregates based on the dominant nature, dominant alleles of the two different characters segregating together, and the recessive alleles of the two different characters segregating together. Or the other hypothesis was the independent assortment, where each allele for the different character is independent from one another. So the allele for color is totally independent from the allele for the nature of the seeds. So this could be expressed by a Punnett square, where we have four different possibilities of the gamete. When we consider the parental generation to have yellow round seeds crossed with green wrinkle seeds, the F1 generation had yellow rounded seeds as their dominant phenotype genotype, and they express yellow round seeds as their phenotypes. Based on this, when he crossed the F1 generation, when he self-pollinated or cross-pollinated the F1 generations, he observed there were four different combinations of 3 different combinations of phenotypes, giving rise to four different phenotypes, 3 different combinations of genotype, giving rise to 4 different uh, phenotypes. So based on that, he came up with the conclusion that independent assortment of alleles is the most probable one. Why? Because the offsprings showed 4 different phenotypes, one being yellow and round, the other being yellow and wrinkled, then green and round, and green and wrinkled. If they had been dependent assortment, they cannot form yellow wrinkled plants or green round plants. So since the yellow wrinkled plants and green round offsprings were also visible, he concluded that the independent assortment of alleles is the correct assortment. So considering this, he expressed this in terms of gametes as capital Y simple capital Y the dominant allele for the color, and capital R the dominant allele for the nature of the seed, and also capital Y simple R, capital simple Y, and capital R simple Y simple R. So these were the 4 different types of genotypes of the gametes he came up with. So when the two dominant gametes were crossed, yellow color and round, they gave the phenotype capital Y, capital Y, capital R, capital R. And then this combination would give capital Y, and capital R, simple R. And then this combination would give capital Y, capital R, simple R. And then so on and so forth. So these were the combinations. These were the combinations he was able to observe. These were the different combinations he was able to observe, and these gave the ratio yellow round, 9, and yellow wrinkled, and green round as 3, and green wrinkled one as 1. So these were the ratios he observed. These were the observations. These were the observation he got from his experiments. So based on this, he came up with the second law of inheritance, the law of independent assortment. So here the law states that alleles separate and pair up independently during the formation of gametes. So as we mentioned earlier, the alleles for different characters are separated during the formation of gametes, such that two or more genes assort independently, irrespective of the other. But again, this is only applicable to two circumstances, where one is when the two genes considered are on two different chromosomes, are located on different chromosomes, or the second situation where the two genes are located far apart on the same chromosome. On this, they have to be either on two different chromosomes or on the same chromosome far apart. Again, that is self-explainable. Why? If they were very close by, then the chances of these two genes being dependent on one another is slightly possible. So the second law of inheritance is applicable to two circumstances where the two genes considered are located on different chromosomes, or the two genes considered are located far apart on the same chromosome. And then the success of Mendel's experiment was mainly because Mendel carried experiments in a certain way, certain manner that made his experiments a better success than the experiments of the others. So the features of his experiments were Mendel carried out thousands of genetic crosses of any given kind. So when he considered the flower color, he performed the same test repeatedly thousands of times. And he also did this in a very large sample size. So because of the large sample size, the probability of error reduces. So the main one of the main features of his experiments that allowed his experiments to be a success was that he carried out many experiments. And then another feature of his experiment that allowed the experiments to be successful was that he kept accurate records of his results. So he kept proper records of all the experiments he performed, and he probably had them tabulated or something, so that he was able to observe the trends or the patterns that were there in the experimental findings. So he kept accurate records. And then the third feature that allowed him to come up with his conclusions are that he followed up each cross for at least two offspring generations. So this allowed him to find out more than other experiments, because when we consider the flower plants even if we consider the flower colors of the pea plant, if we had stopped in the first step of the first offspring, let's say they had capital B and capital B, right? So if they had purple, we cross-matched the purple color flowers and the white color flowers. And then the offspring all showed purple colors. So based on this, we wouldn't have known that there are two factors involving in this. We would think that purple is always dominant to white, regardless of the situation. Because he was able to follow up this with another crossbreeding, we were able to find out the ratios and the factors that involve in we were able to come up with the different ratios for the heritable feature, and based on that we could come up with the conclusion that there were two alleles responsible for each of the characters expressed. So this was another major reason for the success of Mendel's experiment. He usually followed up for at least two offspring generations. And then he also did a quantitative analysis. So he always had a count as to how many phenotype is expressed by each of these offsprings. So if you guys, you don't have to remember this, but if you can remember, it was 702 purple color flowers to 224 white color flowers. So from this surface level observation, we won't be able to find out this. But since he was repeating this for several different characters as well, he was able to come up with this theory. And then properties of the garden peas for this genetic experiment were another reason for his success. The fact that he chose this plant was another reason for his success. Why this was more useful as a experimental subject is because pea plants are available in many varieties with contrasting traits. So if we consider the features, there are plants with purple flowers and white flowers. We had green seeds and yellow seeds, round seeds and wrinkle seeds. They had pods with green pods and yellow pods. So there were different varieties with contrasting traits as well. So each of these traits are completely different from one another. So that is called contrasting traits, and there were different varieties. So that was a feature of the garden peas that allowed these experiments to be a success. And then also the generation time for the garden pea plant was short. So he didn't have to, if we consider a mango plant or something, if we took a subject like that, then it would take at least 15 or so years for the plant to grow itself. So imagine a scientist doing an experiment on mango plant. He would, by the time he does, even if he starts as soon as he was born, by the time he gets to the second generation, he would be nearly 30 years. So that means it's kind of hard to come up with observations, right? So the fact that garden peas had short generation time was a major reason for his success. And then also large number of offspring is produced from each cross. So this allowed him to observe the phenotype ratios more accurately. If there were only few offsprings, then he wouldn't be able to observe the phenotype ratios accurately. And then crossing between the plants could be strictly controlled. He was able to control whether they are self-pollinated or cross-pollinated. So that was another reason where the properties of garden peas gave him a better success for his experiments. And then the probability laws and Mendelian inheritance. So Mendel's law of segregation and independent assortment reflect that the same rules of probability applies for genetics as well. That means when we toss a coin, the probability of us getting a head or tail is always half, right? Similarly, the probability of one of the probability of each egg carrying a dominant allele from the F1 during the allele segregation in an F1 plant. So the F1 plant is generally a heterozygous plant, heterozygous in a monohybrid cross. If we consider the F1 generation, it's always heterozygous, right? So the probability of the egg containing a dominant allele is also half, and the probability of the egg carrying the recessive allele is also half. So this is something like, if we consider the flower color, the parental generation has capital B, and simple B, simple B. The F1 generation would have capital B, simple B. All the F1 generation would have the same genotype, capital B, simple B. So when this plant forms the gametes, it can only form capital B or simple B, right? So the probability of this forming capital B and simple B is equal, right? It's equal, but when they are forming an offspring, the probability of the character being expressed could be varying. So here, the probability of the allele having, the gamete having the dominant allele or the recessive allele is equal half and half in this situation, in a monohybrid cross. And also, when we consider the probabilities of all possible outcomes in an event, they add up to 1. So this is also another probability, rule of probability. So if we consider an egg having a dominant allele, the probability of the egg having a dominant allele would be half, and the egg having recessive allele is half. And so when we consider them to be, when we consider all the possible outcomes, the probability is going to be 1. So if we consider these two rules for a dihybrid F1 generation of a dihybrid cross, so if we consider yellow round seeds, the genotype would be something like this. So the probability of the gamete having yellow, the dominant allele for the color and the dominant allele for the dominant allele for the dominant allele for the nature of the seed is going to be less than half. It's going to be 1 over 60, 1 over 4. So another probability law is that when the occurrence of an event does not affect the occurrence of another event, so something like mutually exclusive events, you guys would have studied in O levels. So the probability of the simultaneous occurrence of both events can be obtained by multiplying the probability of one event by the probability of the other event. This is known as the multiplication rule or the product rule in probability. So if we consider the F1 generation of dihybrid cross, we have yellow rounded seeds, but the genotype is going to be something like this. So the probability of this seed having yellow allele, the dominant allele for the color, is one exclusive event, and the probability of this gamete having dominant allele for the rounded seeds, rounded nature of the seed, is another exclusive event. So the probability of having yellow color allele and the rounded nature of the allele on the same gamete can be found out by multiplying the probability of each of these individual events. So the probability of yellow color being there is going to be half. Why? Because there are only two possible outcomes, right? The gamete can either have yellow color, the dominant allele, or the recessive allele. If it has the dominant allele, then we consider that probability as so they both have half probability. So this is the gamete combination we want, that we are considering. So the probability of dominant allele for the probability of the dominant allele being in the gamete is half. And then when we consider the nature of the seeds, they are independent of the color. So regardless of whether it's capital Y or simple Y, when we consider the nature alone, they could be either only rounded or wrinkled. So if it's round, the probability of it being rounded is half. It having the allele for rounded nature is half, and the probability of it having the allele for wrinkled nature is also half. So that the probability of nature of the seed is also half. So this is a mutually exclusive event to this one. So the probability of both of them occurring together can be found out using multiplying the individual probability of occurrence. So the probability of occurrence of dominant allele for color is half, and the probability of dominant allele for nature of the seed appearing is also half. So when we multiply this, we get 1 over 4. So this is the probability of us getting the dominant alleles for both the features. And similarly, we can also consider for dominant dominant allele for one feature and recessive allele for another feature. Again, how do we do this? We have to find out the probability of this one and then multiply it by the probability of this one. So the probability of capital Y being there is half, and the probability of simple y r being there is also half. So capital Y is half and simple r is half. Half into half, the probability of capital Y simple r being as the allelic combination of the gamete is going to be 1/4. And then another um, another probability law is that that any one of the two or more mutually exclusive events will occur is calculated by adding their individual probabilities. This is known as the addition rule or the sum rule. So this means when we consider an egg, when we consider gamete having the dominant allele coming from the egg and recessive allele from the sperm, when we consider the gamete, there could be two possibilities, right? The offspring is going to be capital Y, simple Y. The genotype of the offspring is going to be capital Y, simple Y, and capital R, simple R. So the gamete combinations are going to be capital Y, capital Y, simple R, simple Y, capital R, and simple Y, simple R. So the probability of the gamete having the probability of offspring having yellow color and wrinkled rounded nature, yellow color and rounded nature, that could be three different types of phenotypes where capital Y and capital Y, these two this allele could come up with this or this allele, or I think it will be better if we go with a different allele, different example. If we consider the probability of the offspring having green color the probability of the offspring having green color the probability of the offspring having green color and wrinkled nature is going to be from green color and wrinkled nature could be from capital C, capital G, sorry. The probability of having a heterozygote in the F2 generation could be from two heterozygous genotype in the F2 generation could be possible due to two different genotypes. One could be either capital Y, simple Y, or of a monohybrid cross. If we consider a monohybrid cross, the probability of having a heterozygote genotype in the F2 generation could be possible due to two different genotypes. One could be either capital Y, simple Y, or of a monohybrid cross. If we consider a monohybrid cross, if we consider a monohybrid cross, if we consider a monohybrid cross, the offspring, the F2 generation having a heterozygous nature is probable by two probabilities. One is the egg having capital B and the sperm having simple b, or another occurrence is possible where the egg has simple b and the sperm has the capital B. So both of these would have individual half probability, and that would result in 104 probability, and that would add up to form the total probability of half. So if I go over this again, the probability of any one or two or more mutual exclusive events occurring is calculated by adding their individual probabilities. So if I was to represent this in a Punnett square, so these are the alleles from the egg, and these are the alleles from the sperm. So the F1 generation both have this genotype. So the alleles would be capital B simple b and capital B simple b, and their genotype would be something like this. The probability of this offspring having the phenotype, the heterozygous genotype, is probable through two different occurrences, right? So the first occurrence would be where the egg will have simple b and the sperm would have the capital B. The second occurrence is when the sperm has simple b and the egg has simple b. So the first probability where this egg has simple b and the sperm has capital B is 104. And the probability of the egg having capital B and the sperm having simple b is 104. So the occurrence of heterozygous event, which is mutually exclusive, right? The probability of this coming from this combination is mutually exclusive to the probability of this coming from this combination, so that the overall probability of that happening is we can find out using adding them both. So the overall probability is going to be half. And then in multifactorial crosses, where we have two or more characters of an organism are traced, to simplify the nature, we can, using a Punnett square could be complicated. So to apply rules of probability is easier. So again, according to the law of segregation, a multifactorial cross can be considered equal to multiple independent monohybrid crosses occurring simultaneously. So if we consider flower color and pod color, the seed color, then we can consider this to be an individual cross and this to be a individual cross, so that these two are mutually exclusive events, and they could either be found, the result could be either found out using the sum rule or the multiplication rule of the probability. So when we consider dihybrid cross for seed color and seed shape, when we consider a dihybrid cross for seed color and seed shape, the probability of the seed color based on the probability of the seed color is going to be something like this. This would be the Punnett square for that. capital Y, simple Y, so that the probability of seed type is going to, seed color is going to be capital Y simple Y and capital Y simple Y of the F1 generation. So these are the probabilities. We can sum it up as the genotype being possible as 104 and capital Y being 104 and capital Y simple Y being 102. This 102 is from the addition of this 104, 104 and this 104. And then simple Y would give us 104 again. So these are the probability of the genotype, this genotype to occur, and the probability of this genotype to occur is half, and the probability of this genotype to occur is 104. And then similarly, when we consider the seed shape, the probability of capital R occurring is 104, and the probability of capital R simple R occurring is 1/2, 1/2, and the probability of simple R forming is 1/4. So these are two mutually exclusive events. So to find out the occurrence of a heterozygote, a dominant allele for black color, a dominant allele for yellow color seed, and recessive allele for the probability of occurrence of yellow color seed is going to be the addition of these two, right? This is an individual event. This is an individual event. So that's going to be half plus 1/4. And the probability of a round seed to be occurring is the addition of these two. This is going to be 3/4. And the probability of round seeds from forming is going to be, again, 1/2 plus 1/4. That's going to be 3/4. And the probability of us having yellow color round seeds is going to be the probability of this occurring times the probability of this occurring. So 3/4 times 3/4. So the probability of yellow color rounded seeds occurring is 9/60. Or if we simplify this, it's going to be, we can simplify this, 9/60. So again, this is the ratio we obtain from the Punnett square as well, right? I believe in the dihybrid cross. In the dihybrid cross, when we go with capital Y's, when we come across, when we consider different genotypes, the genotypes could be capital Y, capital Y, and capital R, and capital Y, and capital R, simple R, and so on and so forth. So the probability of yellow color round seeds occurring is 9/60. And if we consider another feature where we can consider green color round seeds, then the probability of green color to occur is going to be 1/4, and the probability of yellow round seeds to occur is 3/4, and the probability of green color round seeds to occur is going to be 3/60. And then the probability of yellow color wrinkle seeds to occur is also similar, where we have the probability of yellow color occurring is 3/4, and the probability of wrinkle seeds to occur is 1/4 from this, and the probability of yellow color wrinkle seeds to occur is 3/16. And then the probability of yellow green color wrinkle seeds to occur is going to be, the probability of green color to occur is 1/4, and the probability of wrinkle seeds to occur is 1/4. That's going to give us 1/16, a probability. So when we observe the offsprings of the Mendel's second dihybrid cross, he observed that yellow round seeds, and then yellow wrinkle seeds, and green round seeds, and green wrinkle seeds in the ratio 9:3:3:1. So this is the phenotype ratio we observe in the Mendel's dihybrid cross. So when we put this into a probability chart, we can get the same results. And then we have something called the test cross. The test cross, if you guys can remember, the test cross is a deliberate breeding process performed in order to find the unknown genotype using the genotype of the offspring. So the genotype of an, when the offspring, when the parental generation, breeding of an organism having unknown genotype for a specific dominant trait with an organism having homozygous recessive condition for the same specific trait to identify the unknown genotype. So if we assume, if we perform this during a monohybrid cross, then it's called a monohybrid test cross. And if this is performed during a dihybrid test cross, we call this a dihybrid test cross. So in the probability of when we do a simple monohybrid test cross, we can consider a feature such as tall plants being crossed with the short plants. So the tall plants would have a genotype as capital T, and short plants as simple T, and the gametes would be capital T, simple T. So the offspring is going to be capital T, simple T. Whereas if this was done with a heterozygous plant and a short plant, the gametes form would be capital T, simple T, and simple T, so that the offsprings could be of two different types. Either they could be capital T, simple T, or simple T, simple T. If they were capital T, if they were of the genotype capital T, simple T, then they would be tall. And if they were simple T, they would be short. But if the phenotype was capital T, homozygous dominant, and homozygous dominant, the offspring would only be tall. So if we have an unknown tall plant, a tall plant with an unknown genotype, if we cross it with a recessive short plant, and if we get, based on the offsprings we can identify the genotype of the parental plant. So if we get only tall plants as offspring, then the genotype of the parental plant is going to be capital T, the dominant homozygous dominant genotype. Whereas if we get both tall plants and short plants in equal proportions, then we can say the genotype of the parental plant would have been capital T, simple T, or heterozygous dominant genotype. So this is how we observe this is how we identify an unknown genotype of the parental plants using a test cross for monohybrid cross. So when we consider dihybrid cross, there are two features to be considered. We can come up with we can come up with the parental genotype based on the offspring phenotype. So for this, if we consider an example where where we cross plants having red color flowers with black color seeds, this is the dominant dominant homozygous. And then the recessive trait would be white color flowers with brown color seeds. So the dominant feature would be red and black color seeds. The recessive feature would be white color flowers and brown color seeds. So when we have a red color flower and brown color black color seed giving plant, we don't know the genotype. So what we do is we cross it with the recessive, with a plant that shows a recessive feature for both. So the genotype of the recessive plant is going to be simple R, if we consider this to be capital R, and capital B, and this to be simple R, and simple B. We cross it with a plant having the genotype simple R, and simple B. If it had the phenotype, this type of dominant nature for both of these characters, homozygous dominant nature for both of these features, let's say capital R, and capital B, the offsprings would be, these are the gametes formed. The gametes form would be capital R, capital B, and simple R, simple B. So the offsprings form would be, all of them would be capital R, capital B, simple B. This would be the genotype of all the offspring. So if they had red and black, if they had this phenotype, the parental plant has this genotype, then the offspring would definitely show this phenotype, where it shows the phenotype of red color flowers and black color seeds. So when we cross a parental plant of unknown genotype showing red color flowers and black color seeds with a homozygous recessive nature for both the features, and the offspring is also showing dominant phenotype for both the features, then we can say that the genotype of the parental plant is also going to be homozygous dominant. And when we consider the other situation where this could be homozygous for one feature and heterozygous for another feature. It could be homozygous dominant for red color flower, or it could be heterozygous for the red color flower color, while being it could be dominant for red color flower color, homozygous dominant for red color flower color, and heterozygous for seed color, or it could be heterozygous for flower color and homozygous for seed color. We can cross it with the homozygous recessive plant for both the features. And the offspring of this combination would be, the alleles formed would be RB capital B and capital R simple B, and this would always be this. So the offsprings probable are going to be this kind of genotype, which would be observable as red flowers and black seeds. And the other genotype possible is going to be capital R and simple B. So this would be red color flowers and brown color seeds. So if we get such an observation, we can say that this feature, the feature that shows both the characters in the offspring, is going to be heterozygous in the parental plant. And similarly, if we consider the flower color being heterozygous, the gametes would have the allelic combinations like this. So that the offspring genotype is going to be something like this. Here, this would be red color, black seeds, and this would show white color, black seeds. This feature would be express this feature would give us the information that parental genotype is going to be heterozygous for that nature, heterozygous for that character. So the feature that shows two different offspring phenotypes will have the heterozygous genotype. And the probability, and in the rare case where both of these are going to be heterozygous, where we have this kind of a genotype combination, when we cross it with a homozygous recessive plant, the offsprings, the gametes would be capital R. This kind, the gamete combinations could be different, like this. So that there will be four different genotypes occurring in the F1 generation. So the phenotype offering a genotype occurring from this combination would be capital R simple R, capital B simple B. This would give red color flowers and black color seeds. And this combination would give us white flowers with black seeds. And this combination would give us red flowers with black seeds. And this combination would give us red flowers with brown seeds. Sorry, this one should be red color flowers with brown seeds. So if all four different phenotypes are expressed in the offspring generation, we could say that the parental genotype is heterozygous for both the features. So this is how we perform a test cross for dihybrid features. This is how we perform a dihybrid test cross to identify the parental genotype. Once again, if it's a monohybrid test cross, the offspring could either show two different phenotypes. One where all the offsprings have display the dominant features only, and another situation where there are 50-50. There are 50% of the offspring show dominant, while the 50% show the recessive nature. For the monohybrid test cross, if we perform a monohybrid test cross and the offspring generation shows entirely dominant phenotype, then we can say that the parental phenotype was also dominant, homozygous dominant. The parental genotype was homozygous. And if the offspring generation showed dominant and recessive offsprings, dominant and recessive phenotypes in equal proportions, then we can say that the parental genotype was heterozygous for monohybrid cross. For a dihybrid cross, we can say that whichever feature shows dominant, whichever character we are considering is showing, if the offspring shows only dominant phenotype for the offspring generation, then that would be that genotype combination of the parental organism is going to be homozygous. Whereas if the offspring is showing both the phenotypes for the offspring for that specific character, then that specific character is heterozygous in the parental genotype. That's about test cross. And then we can come across inheritance of Mendelian characters in humans. So we have some common features in common Mendelian characters in humans as well, where they are the inheritance of the character is dependent on based on Mendelian laws. So this could be either ability to roll the tongue or non-rolling, where the ability to roll the tongue is a dominant feature, while being unable to roll is going to be recessive. And then also the probability of the feature having free hanging earlobe and attached earlobe is another Mendelian feature, where the free hanging earlobe is a dominant feature and attached earlobe is a recessive feature. And then dimples on cheek is another feature that is inherited by Mendelian laws, where the having dimples is a dominant feature, while not having dimples is a recessive feature. And then bent thumb, or what we call as the hitchhiker's thumb, the dominant feature is the hitchhiker's thumb, and the recessive nature is the straight thumb. And also pedigree, and then we come across pedigree analysis. So we have a method of analysis called the pedigree analysis, pedigree charts. So in pedigree charts, there are certain symbols we use to represent certain things. So we have squares that represent males and circles that represent females. And then the non-shaded squares and circles represent unaffected males and females, whereas the shaded ones would represent the affected male or female respectively. And then a link is shown when there is a crossing occurring, and then the offspring is shown by small arrows in this manner. So the first offspring is given on the left hand corner, something like this. So this is the information we need to analyze a pedigree chart. So again, if we consider a common the offspring, the offsprings could show two different phenotypes where they have the widow's peak or they don't have the widow's peak. So this would be an affected female, while this is a non-affected males, and this would be represented again as a affected male. So the genotype of this person is going to be capital W and simple W, and this person is going to be simple W, simple W, simple W, simple W, and this is going to be capital W, simple W again. So why can't this shaded person, these two shaded people can't be capital W? Because this parental gene, only the affecting the allele for the widow's peak is only present in the paternal side. The maternal side does not have the allele. So the only probable way for the offspring to get the widow's peak is through the paternal side. So there could only be one W in the offspring. So in case if they have the simple W from the paternal side as well, then the offspring would show, the offspring will not show pedosp. So this is going to be the first generation, and this one is going to be the second generation. So in case this person gets married to another person who is also affected, similarly affected, has the similar genotype, then the offspring of these two people could be, again, various different kinds. Where they could be affected or not affected. If they are affected, they could be capital W, or they could be capital W and simple W. Or if they are not affected, they are definitely going to be simple W, simple W. Again, the affected type is going to be, the affected type could have got the capital W from the paternal side or the maternal side, or both the side in this case. In this case, it could be either from the paternal side or the maternal side, while getting the simple W from the other side. So similarly, if we consider another example, example for attached earlobe. Here, having a free earlobe is the dominant trait. So we consider two individuals and the offsprings having this kind of a phenotype, phenotype. And then so from this, we can say that from this, by observing this pedigree chart, we know that if they have an attached earlobe, they are represented by shaded part. While if they have a free earlobe, they are going to be not shaded. So this could be either completely free or simple, capital F and simple F. While this one also could be either of the genotypes. But this one is definitely going to be the recessive genotype. Why? Because if they are going to be affected, they have to have both the alleles. Because having attached earlobe is a recessive feature. So these two and this one, these ones are going to be definitely simple F, simple F. They are expressed because they have simple, they have recessive genotype. So it has to be homozygous recessive in order to be expressed. So based on this, we can say that if this has to occur and they both are if the offsprings are showing, if the offspring are showing a recessive feature, then if both the parental types are only having the homozygous genotype for the feature, means they won't be able to have such a child, right? So if only this person is also homozygous and this person is also homozygous, means they won't be able to have this kind of an offspring. In case if one of them has the other, the recessive nature for this, one has capital F, and the other has capital F, simple F, again, this won't be expressed. Why? Because this one has capital F, and this one has capital F and simple F. The possible genotypes would be capital F and capital F, or capital F and simple F. So this is also free earlobe. This is also free earlobe. So this kind of genotype is not possible. This kind of phenotype is not possible. So in order for this phenotype to occur, both of these parents should have had capital F and simple F. Why can't they be simple F, simple F? If they had simple F genotype, then they would also be affected. So since they are not affected, they have to be capital F, simple F. They have to be heterozygous for them to not show the character, but for the offspring to show the character. Similarly, we can say that this offspring could either be capital F, or capital F, simple F. We would not know the genotype unless we consider the offspring of this person as well. And similarly, if we consider this offspring, this person could have capital F, or capital F, simple F. And also this the maternal side from this one. So again, when we consider this person, they could either be capital F, or capital F, simple F. But when we consider the parental genotypes of this person, the paternal side has simple F, and the mother could either have capital F, or capital F, simple F. But since they have another offspring that shows simple F, this mother cannot be capital F. Why? If the mother was capital F, the only possible offspring would, the genotype of the offspring would be capital F, simple F. So in this case, this would be not expressed, so that this would be not possible. So because this offspring has this offspring is showing a recessive nature, we can come to the conclusion that mother has the genotype capital F and simple F. So if the mother has capital F and simple F, and the father has capital simple F, we can come to the conclusion that this offspring can definitely not have capital F. Why? The father does not have a capital F. So this person's genotype is definitely going to be a capital F and a simple F. Why not a simple F and simple F? If they were simple F and simple F, then they would be affected as well. So this has to be capital F, simple F. So since we know the genotype of this person, we don't know the genotype of this person. By considering the genotype of the offsprings, they have an offspring with simple F, which means this person also should be capital M, capital F, simple F. I hope this is clear. And again, the phenotype of this person could be capital F, simple F, or capital F, capital F. If we consider another example, where we consider the ability to roll tongue, where the ability to rolling tongue is considered as affected. This is the dominant trait as well. So if we have a pedigree chart like this, you have a pedigree chart like this. We can say that since rolling the ability to roll tongue is a dominant feature, we won't be able to identify the genotype of these people. But we can say that the genotype of these people, whereas we can say that the genotype of these people are going to be definitely simpler. Why? Because if they can only express the recessive feature if they are homozygous. So we know the genotype of all these people, and using this knowledge, we can come with the possible genotype for these people as well. So since this person is having the ability to roll the tongue, he could either have this combination or this combination of the genotypes. But if he had this genotype and then his wife has this genotype, then the offspring is not possible to have this genotype. So in order for the offspring to have this, show the recessive nature, he should be heterozygous. And similarly, we won't be able to identify this offspring, whether it's capital R simple R or simple R simple R. And this person, we won't be able to identify as capital R simple R, okay, simple R simple R, capital R. And this person also, we don't know the genotype, whether if it's capital R or capital R simple R. But by considering the parental genotype has simple R simple R. So this has to be capital R simple R. And again, this parental genotype also has simple R simple R. So again, the probability of capital R being is eliminated. I think I made a mistake here. It can only be capital R simple R. The simple R probability is not even there. The capital R is the probability, but because of the parental genotype, we know that's not possible. So because capital R and simple R is the genotypes, this person could have simple R, but this person could either have capital R as genotype or capital R simple R. And then since we know that this offspring is possible, we can say that genotype of this person has to be capital R simple R. And then the chance of having video speak can be identified using multiplication rule as the chances of having rolling tongue can be found out using the multiplication rule as well. So here if we consider this with another feature as well, such as video speak, where video speak is the recessive nature, video speak is a dominant nature, we can say that where we consider attached earlobe, where attached earlobe is the recessive nature, we can say that the chances of having both the ability to roll the tongue and attached earlobe is going to be the chances of having both the ability to roll the tongue and having attached earlobe is going to be the multiplication of the probability of the probability of this one and the probability of this one. The probability of this in a dihybrid cross of F2 generation is going to be 3/4, and the probability of this one is going to be 1/4. So the probability of having both the features is going to be 3/16. So so based on this, we can say these are the features of Mendelian inheritance. Inshallah, we'll discuss about the non-Mendelian inheritance in a different seminar. You can ask any questions you have. Someone had asked to explain me again. Sorry, I missed your message. Can you tell me which part of the lesson so that I can explain it again? Okay. So if you have any questions, you can ask me now. If you guys don't have any questions, then I would like to conclude the meeting. Thank you for, yeah, inshallah, there will be another session. We will be informing you through the same way you got to know about this message, I guess. Yeah, and in that case, thank you everyone for joining this session. Hope to meet you in the next meeting. And you're welcome.